Hello everyone. The topic which I am going to cover up in this video, it is development of Dicot embryo. But before we could start with the development of Dicot embryo, let's just go for a quick short revision of double fertilization and triple fusion. So over here, let's talk about the first, that is double fertilization. Fine. This is an event which is exclusively found in the angiosperms, right? And in this, as we have studied earlier, that when there is a germination of pollen tube on the stigma, just like this, right? Let's consider this as style, and here you have stigma. We have, that is, a pollen grain over here. So when there is a germination of pollen tube on the pollen grain, on the stigma, right, there is two male gametes, right? We have how many male gametes? That is two male gametes which are being forwarded towards the ovule. Now, what happened in the process of double fertilization when this pollen tube enters inside the ovule? Fine. There are two male gametes which are released in the ovule or the embryonic sac, I must say, to be on precise. So what is going to happen over here is one male gamete which is an in condition, right? One male gamete which is an in condition fuses with egg cell, which is again haploid in condition, give rise to something called as zygote, which is 2N in condition. And we are familiar, this process is called as fertilization, or we can call it as syngamy. In either of the ways, which are synonyms, usually which we prefer over here. Now the second part which comes is the another male gamete. The another male gamete, which is 2N, which is N, that is haploid in condition, fuses with a cell which is present on the center of the embryonic sac, that is called as the central cell. Now, what is special about the central cell is that the cell has two nuclei, therefore the condition becomes N plus N, that is a dikaryon condition. Now, N plus N plus N, then it gives rise to a triploid condition, which is somewhat called as primary endospermic nucleus, which leads to the development. And now this primary endospermic nucleus will develop into the endosperm, which leads to the development in the endosperm. Now over here, so these two events, that is two fertilization, are taking place. That's why these two are called as double fertilization. Whereas if you can see over here, there are three nuclei which are coming together to form a triploid cell. This is called as triple fusion. Fine. So this is what we are going to study. We are going to study the development of endosperm later on. First and the foremost, we are going to study what happens to the zygote which is present over here. Now, when I am talking about angiosperm usually have two fates. Either the zygote will turn into a monocot, or either it will turn into a dicot. Fine. So let's talk about, right now in this video, we will be exclusively talking about this zygote turning into a dicot embryo. So when I am talking about a dicot embryo development, fine. First and the foremost thing, you must know that when I am talking about dicot embryo, this type of development is called as cruciferous type of development, which is most common in the dicot, and then comes the meroplastic type of development. Meroplastic type of development. Fine. So these 2 points are to be kept in mind, that is cruciferous, and then comes the meroplastic development, and these two terms will be explained while we will go deeper into the topic. So just keep these two terms in mind, that is crucifer, and then comes the meroplastic type of development. Now let's start with the development of that is zygote into a dicot embryo. So when I am talking about a zygote is a 2N condition which is being developed after the production of that is the product of fertilization after the formation of after the fusion of male and female gametes. So over here, for the first, what is going to happen? We have a zygote. Fine, we have a zygote which is 2N in condition. Now, first, before the zygote starts dividing, that means developing into an embryo, first, zygote will eat up the nutrition which is present in the embryo sac, raises to the maximum size, means it is increasing its size and shape and the content before it could divide. So the first step is zygote absorbs nutrient. Fine. As the zygote absorbs the nutrient, it will get larger in the size and the resultant structure is called as oospore. The resultant structure is called as oospore. Zygote absorbs the nutrition, develops into the oospore. Now, this oospore is going to go for the first division. Then, the first division is transverse, right? The first division is transverse division. That means one on the other. So, you get two cells over here, one on other. Now, for understanding the location of these two cells and their fate, we must recollect our structure of an ovule. So, let me just roughly draw a structure of ovule over here. Let's consider this as an ovule, outer integuments, inner integuments, and the embryonic sac. You are familiar with this structure. This part is your chalaza. We know that very well. This part is your micropyle region. Fine, this is your micropyle, that is your chalazal region. And this division is taking place over here. So you are getting two cells, one on the above. One on the above. I donate this two condition with one is called as the C over here for the chalaza and M over here for the micropylar region. Now, what happens to them? Now, these two cells, the one cell which is towards the chalazal side, it is called as the apical cell or embryo cell. Fine, you can call it as apical cell or the embryo cell. So the one over here is your apical or embryo cell. Fine, let's go for the next one. The one which is towards the micropyle, that is the lower cell over here, right? It is referred to as the basal or suspensor cell. Fine. It is referred to as the basal cell or the suspensor cell. Let me write it over here also. So this is your basal cell or suspensor cell. Fine. So this is the two structures which you get over here. Fine. So basal cells or the suspensors towards the micropyle. Then you have the apical embryonic cells which are towards the chalazal side. So I hope you are clear onto their positions now. I will be denoting them as C and M as the direction of chalaza and direction of micropyle over here. Now let's go to the first division. Now once they are divided, now comes the second division. In the second division, embryonic cells, they are dividing spontaneously. Continuously they are dividing on their own slowly, whereas suspensor will always divide transversely. Suspensor transversely, that is one on the another in a straight chain like structure. Fine. So what is going to happen in the second step? This was step 1, 2 and here comes the step 3. embryo cells are going to divide, so 1 and 2. 2 cells will be from 1 you get 2 cells. Then basis, as it is transverse, you will get 2 more cells over here. Now, if we see this structure, it resembles a T. This is called as T-shaped embryo. Fine. This is called as a T-shaped embryo. Fine. Let's move to the second part. Okay. Now, after the T-cell embryo, you will have what? That is the embryo cells which are above. They are again dividing, and the suspensor cells are again dividing. So you get a structure something of this kind. That means the embryo is going to form a tetrad over here, whereas the suspensor 1, 2, 3, 4, 5, 6, and like a big cell. So it is going to form a long chain-like structure of 6 to 10 cells depending on the plant. 6 to 10 cells, right? 6 to 10 cell structure, right? And it is called as hypophysis, right? This is called as hypophysis. And the cell at the bottom, that means the last cell, it will get swollen up, a bit bulged up, and it is called as hystrical cell. Fine, it is called as hystrical cell or hystrical cell. Fine, is it clear over here? Right? The embryo is going to form a tetrad, whereas the 6 to 10 layer long cell chain is called as hypophysis, and then the last one will develop into a bulged structure that is called as hystrical cell. Now, what is the function of this hypophysis? Now, when I am seeing the development of hypophysis, so let me just rub this and just interpret it in a better way. Fine. So let's study the structure over here in this diagram. Fine. So we have a hystrical cell over here, and you have various suspender cells, and here you can see the tetrad which is being developed over here. Fine. So as this region is called as hypophysis, and this is your hystrical cell. Now you can see this. This is your this is your, what I called it as your tetrad, and that is your embryonic cells. They are being pushed towards the center. If you see, they are being pushed towards the center because center contains nutrition. So the embryo has to be pushed above so that the embryo can absorb the nutrition. It can use that nutrition for the better development of the cells. Now, this is to be understand that is the hypophysis has one function, that is to put the embryo above. Towards the chalaza, they have to push it towards the center. To more precise, to get more nutrition, to go for a better development. Now, let's go for the second type of development. Now, once this development is completed, now the second set of division starts. That means out of 4 cells, you get 8 cells. Now, you are going to get what? A 8-celled condition. And again, hypophysis will remain intact over here, right? As usual, hystrical cell you have, and here you have the hypophysis. Now, this is an octameric condition, right? This is an octamer and this is the tetrad condition. Fine. So when I am talking about this condition, you get 2 layers of cell above and below. The one which is just next to the hypophysis, it is called as hypobasal cells. The one which is near to the hypophysis is called as hypobasal cell, and the one which is right above the hypobasal is called as epibasal cells. They are called as epibasal cell. Now, they have their equal importance, just like hypobasal will help in the development of roots, whereas the epibasal will help in the development of shoot. Now, let's move further. Again, they will be dividing, that means third, fourth, fifth, sixth structure over here. Now, as it is dividing after, so we get usually in any of the cell division 1 to 2, 2 to 4, 4 to 8, 8 to 16 cell. Now the condition is a 16th cell or a 16 cell condition over here. Fine. So something of this kind you are going to observe, right? And again, hypophysis on the same position. Can you see this structure? This is a 16 cell condition. So it is a 16 celled globular embryo, or we can call it as proembryo. This is to be remembered by you precisely. 16 cell circular globular structure of, that is, embryo, considered to be as proembryo. Now, after globular, because of the continuous division, and we are talking about dicotyledons, now slowly and steadily their development will start. So, after this, you get something similar to heart-shaped structure over here. Fine. Something, the embryonical cells will develop a heart-shaped structure over here for the continuous division and the formation of cotyledons. So, we get a heart-shaped structure. Again, the hypophysis remains the same over here, pushing the embryo towards the center. Now, this is a heart-shaped structure and we call it as heart-shaped embryo. Right? We call this as heart-shaped embryo. Fine. Just you can see now the variations over here. It started from a single cell to 2 to T-shaped, tetrad, octamer, to a 16 cell globular, to a heart-shaped division. Now what happens is, now these edges are continuously dividing, and as they divide they form the cotyledon, and as they form the cotyledon, what happens is the rate of division is so high that the cotyledons start curving. That means they will start bending towards the other side. So this leads to the formation of some structure which is similar to this kind. Some structure which is just like this, and this will be completely filled with the cells over here. And this is resultant of the continuous and the fast rapidly dividing structures of the embryonic cell. So over here, this is what we get. Fine. I hope you are understanding what is happening over here. So this is what the structure we get. The structure after the heart-shaped embryo, this is a Dicot Embryo. Now, what happens is, let me tell you the structures. These two elongated structures are called as cotyledon. Fine. The position where this cotyledon combines is what we call as plumule. Right? The part which is next to that of your hypophysis is going to develop into a thread. So, this is how the embryo starts developing. Now what is going to happen once the embryo is completely developed? There is no use of this hypophysis and the suspensor. And as there is no use of hypophysis and suspensor, they are going to go for disintegration. Disintegration means they will be completely degraded and removed from, and then you get the final structure of the embryo, which is something of this kind. So this is what the structure of Dicot embryo looks like. Now let's compare it with the formation of a imaginary embryonic axis. That means a line which is passing to the center of the embryo. So I will just go for drawing a dotted line over here, and this dotted line is considered to be as the embryonic axis, and we can call it as tagellum over here. Fine, this embryonic axis is called as tigellum or tegellum, which is found over here. Again, see this two structure, cotyledon. Fine. The point where this cotyledon fuse, they are going to form the plumule. Fine. Right. On the exact opposite, that means exact opposite side, you are going to get the radicle. Right. And as you know that radicle are protected by a cap-like structure called as root cap. So here you are going to have a root cap. Fine. And then the two structures which are left, that is epicotyle and hypocotyle. The region of tissues above radicle, hypocotyle, and the region after plumule is called as the epicotyle. So this completes the structure of a complete dicotyledon embryo, and this is the stepwise development of them. So 6, 7, 8, and the last one 9. Fine. So I hope you are able to understand. Let's go for a quick revision. Zygote absorbs the nutrition, forms the oospores, transfers division to cells. On the chalazal side, apical or embryonic, micropylar side, you have suspensors. Now, suspensors go for the division, embryonic goes for the division, leads to the formation of a T-shaped structure. Then you get a tetrad and a formation of hypophysis. And the last part of the suspender goes development with the gastrical cells. They again go for the development, forming a two-layered structure. The one which is associated with the hypophysis, hypobasal cell, and the one which are above it, epibasal. Epibasal develops into the plumule, hypobasal develop into the radicle. Now, they again go for the division, forming a 16-cell structure, a globular structure, which is called as polyembryo. Polyembryo develops into the heart-shaped structure. This heart-shaped structure continues to rapidly divide, continuously divides to form what? That is a dicot embryo, which you can see two cotyledon where they fuse together gives you the plumule, and the opposite direction the radicle. Now, no need of suspensor, degraded. After degradation, this is how your embryo will look like. So, two cotyledon, embryonic axis called as tegellum, then the point where they fuse, plumule, region after plumule, epicotyle, hypocotyle, radicle, and the root cap. So this is the complete structure of your dicot embryo, which is being mentioned by me in a single stretch. So please note it down, it will be helpful for you. Thank you.